Dr. Praveen?
Sir, can you hear me?
Yeah. You're there?
Okay. Yeah, I'm here.
You're ready?
Yeah.
Okay. Shall we start, madam? Madam, you can. Good evening, everyone. Warm greetings to all. Welcome to today's international webinar, Cancer Immunotherapy, a paradigm shift in the treatment of cancer by Dr. Praveen K. Bommareddy, Associate Director, Preclinical Immunology, Replimune, United States of America. Before we start the webinar, I would like to share a few points about the host and steering committee. I'm glad to host today's session, and I, Dr. S. Ramchandran, Vice Principal and Head of the Department of Pharmacology from GIET School of Pharmacy, Rajahmundry. Three Kaundinya Educational Society, founded GIET School of Pharmacy as a part of GIET institutions by founder chairman Dr. KVV Satyanarayana Raju and Vice Chairman Sri K. Sasikiran Verma in the year 2004 with introduction of different pharmacy courses with a gist for spreading quality in healthcare education. The institute itself has a number of laurels for its splendid achievement. The eminent head of the institute is Dr. M.D. Dhanaraju, a good administrator, academician, and researcher with 25 years of excellent experience. And today's webinar session is hosted by Department of Pharmacology, established in the year 2010, headed by Dr. S. Ramchandran, myself. The department has grown men and materials, faculty and infrastructure since times. The department has succeeded in finding suitable employment for those who have completed post-graduation. The department has well-experienced, expertised faculty. The Department of Pharmacology guided 75 PG projects and many UG and PharmD research projects and published 65 national and international publications in reputed journals and in conferences. The postgraduates from the department are presently working in many international and national pharmaceutical research organizations, especially in countries like Germany, United States of America, Australia, and so on. The department has well-established CPC SEA approved animal house facility which supports our research. Now, I would like to discuss about the objectives of today's webinar, Cancer Immunotherapy, a new class of cancer therapy, collectively termed immunotherapy, work by enhancing or modifying the immune system to re-establish a targeted elimination of cancer cells. Because of these treatments work by harnessing a patient's own immune response, they have the potential to achieve long-lasting remission with fewer side effects compared with traditional cancer treatments. As immunotherapy is a new checkpoint for all cancer survivals, as the research in this area is quite encouraging, as it is one of the emerging treatments and technologies which keeps the researchers to move forward to the future. Now, before I pass on the session to Dr. Sumalata, co-convener of this session, I would like to share the registration statistics of this webinar. We are extremely happy to share the webinar registration details with you. There is a massive response from faculty and student community for attending this today's webinar. There are total 2,366 responses, and we are extremely thankful to all the faculty and student community for showing interest in this webinar. Now I request Dr. Sumalata to take over the session. Madam, please.
Thank you. Yes, sir. Thank you very much. Hello, everyone. So I am Dr. Sumalata, working as a Vice Principal for Vikas Institute of Pharmaceutical Sciences, and my specialization in field is pharmacology, pharmacology. It is my prime duty to say a few lines about our institute, like where I am working. That is Vikas Institute of Pharmaceutical Sciences. So it is an institution of learning of the highest level with different variety of courses such as graduate and postgraduate, including we have BPharm, PharmD, PharmD, and MPharm. The first major strength of institution is taking possession of building with an area of 2.54 acres with a perspective of bird's eye niche in such a diverse atmosphere. The power that is driven in enlightening came from fostering quality teaching with a range of challenges encapsulated by a number of professors with vast guidance and many research scholars in our institution. The unique golden opportunity of our institution is Dr. T.V. Narayana sir. Presently, he is the president of Indian Pharmaceutical Association, a moving encyclopedia in the field of pharmacy, and he is the backbone of our Vikas Institute of Pharmaceutical Sciences. His vision and strategy with bottom-up experiences and innovation in teaching and learning. So, is a great advantage to our institute. On the other hand, myself, Dr. Sumalata, I have completed my postdoctoral fellow from Science and Engineering Research Board, Government of India. This is an added advantage to foster various insights like in the development of inspirational research content in our institute. Even the eminent and notable faculty with different specializations in our institute are one of the strength in considering our institute as a role model. The prestigious major national events like Indian Pharmaceutical Students Congress, 9th and 10th one, were organized at our institute, like our Vikas Institute of Pharmaceutical Sciences. During these mega events, all the 6 Indian Pharmaceutical Association past presidents has participated for the first time where they are keenly indulged in different like official tasks like them. So that program brought together over 3,000 delegates across the nation to share their experiences and to learn from one another and to debate regarding the current and future pharmacy. Numerous students of our institute got settled in many MNCs. So with a good pay scale, it is also a notable landmark of our institute. So with this brief background of our institution, today really I am very fortunate to be a co-convener for this webinar, so Cancer Immunotherapy. Now I'd like to introduce our today's speaker, very young and dynamic speaker, Dr. Praveen. So he's an Associate Director from Preclinical Immunology and Translational Research, USA. Hello, Praveen. Could you hear me? Yeah, okay. So let me introduce about Dr. Praveen. So he has profound experiences of more than 10 years in basic translational research and greater than 5 years of different expertise in immuno-oncology. And he is a very powerful publication record exceeding 15 manuscripts, so which is including Science Translational Medicine, Journal for Immunotherapy of Cancer, Nature Reviews, Immunology, Journal of Clinical Investigation, Oncoimmunology, and one patent during the last 5 years. He is having exceptional proficiency in the designing and execution of IND, enabling preclinical studies, target discovery, target validation, and biomarker assays to determine the pharmacokinetic and pharmacodynamic properties of cytokines, cell therapies, molecules, and immune checkpoint antibodies, oncolytic viruses, and other IO drug candidates. Coming to his education, he has completed PhD Molecular Biosciences from Rutgers University, New Jersey, and he completed MS Pharmacology and Toxicology from Long Island University, New York, and Bachelor of Pharmacy from our prestigious Andhra University, Visakhapatnam, India. And we are very proud to feel that he is an Indian and who got settled in US. Really, we are proud of you, Dr. Praveen. And coming to his benchmarks, during his PhD, investigated 2 proposed mechanisms of action of EVEC and FDA-approved oncolytic herb simplex virus 1. He designed and executed and completed a project to understand the immunological and cancer cell signaling effects of triple combination oncolytic virus, MAP kinase inhibitors, and anti-PD blockers. Using CRISPR Cas9, he delineated the possible mechanisms of resistance of oncolytic viruses and identified key biomarkers to his credit. And he tremendously constructed a novel second-generation armed oncolytic HSV1 encoding an immunostimulatory cytokine using a bacterial artificial chromosome system. Really, it's a very great achievement of Dr. Praveen. Coming to his professional organizations, he is a member of Society for Immunotherapy of Cancer, and American Society for Pharmacology and Experimental Therapeutics, American Association of Laboratory Animal Science, and American Society of Clinical Oncology. Really, it's a very great Dr. Praveen. Coming to his honors and awards, he bagged a Dean Award from Long Island University in June 2011, and he bagged Exemplary Employees Award from Memorial Sloan Kettering Cancer Center, and he got Special Study Award from Rutgers State University of New Jersey, and he also got Society for Immunotherapy of Cancer, 30 Emerging Leaders in Cancer Immunotherapy, and inaugural Sparkthon participant. So, and to his credit, he is having a patent on combination immunotherapy for melanoma utilizing oncolytic viruses and immune checkpoint inhibitors, provisional patent submitted by Rutgers during May 2018. Really, we feel very privileged and very thankful to Dr. Praveen. So you are here in spite of your busy schedule. Once again, credit goes to GIET School of Pharmacy Management Principal and also the convener, Dr. Ramachandran sir. So I request Dr. Praveen to give his presentation. Thank you, everyone.
Right. Can you see my slides?
Yes, Dr. Praveen, we can see.
Sure. Again, I think Dr. Sumata gave a great introduction of my background and my education and everything. But I do want to stress that. So the most important thing is you understand science at a very basic level. So you can basically do anything. And once you have that understanding, you can really extrapolate that to identify great things and move on. With that being started. So again, thank you so much for inviting me. Thanks for the panel, and I'm extremely privileged to be speaking at this meeting today. So again, I'm Praveen Bamareddy. I have a master's and a PhD, and most of my work is based in cancer immunology during the last 5 years, and I've done very basic molecular research during the last 10 years, leading to different publications, as just Dr. Sumata mentioned. So I originally finished my pharmacy at GIET School of Pharmacy. Then I worked a few years at Memorial Sloan Kettering Cancer Center. So it's the best cancer center in the world, or at least number one or number two. Then I moved to Rutgers University, where I did my PhD in cancer immunology. So during that time, it's a joint PhD degree with Harvard and Mass General Hospital. So this gave me a really good platform to get connected with the leading experts in the field and get trained by them. And I was extremely fortunate to get trained by some of the experts in the field of cancer immunology. And again, you always have to remember your mentors. And with that, I'll start. So like I said, again, all this presentation is based on my sole knowledge and understanding in the field. So don't consider this as a medical advice. I'm an employee of a private company and it owns stocks. So this seminar is given for educational purposes and not relevant for my employment. And again, the presentation may contain images of patients and laboratory animals. So just if anyone is not comfortable, just letting you know ahead. So the definition of cancer, so many of you might already know or kind of know what's the idea or what's the basic definition of cancer. So it's very complicated. So as you can imagine that it's not very easy to treat cancer. It's not one disease. It's not like coronavirus. I'm not saying it's easy to treat coronavirus. It's something new. So it's going to take some time to get the vaccines and different understand the virus, etc. But for example, in cancer, it's a name given to a specific collection of related diseases. It's not just one thing. So you cannot say mutation of one gene leads to cancer. No, it's a complicated, very complicated thing where cancer has adapted different mechanisms so it can grow and metastasize and eventually spread through the body and live happily. So again, this is the definition of cancer from the NCI. So NCI is the National Cancer Institute in the US. So they provide funding for cancer research and they provide guidelines on cancer treatment, and again, this the leading institution in the country, supported by the U.S. Federal government. So again, cancer is a collection of related diseases where some of the body cells, not all cells, some of the body cells begin to divide without stopping and spread to the surrounding tissue, and sometimes not just surrounding, even to distant tissue. So you have a cancer and your on the skin can metastasize to your lung or to your brain. So it can really, really metastasize and go to distant places and cause significant damage. So coming to the very basics, I just mentioned that unstoppable cell division. So how does this really happen at a normal cell division or at a normal scale? So many of you might already know the cell cycle. I don't want to go into too much detail. So there's a G0, G1, S, and G2 phases, and there's a mitotic phase. So then, end of every phase, these are checkpoints. So these restrict the cell from switching from one phase to the other phase. So the G1 checkpoint, for example, and the G0 checkpoint, the G2 checkpoint, and then finally going to mitosis. So this is a regular cellular processes, and in all normal cells, it occurs with very precision control. But what happens in cancer cells is that you have a loss in these checkpoints due to mutations in genes. So when I say mutation, as you know, the DNA has base pairs, and a deletion or addition or a replacement of one base pair can lead to a mutation. So DNA, RNA, and protein, the central dogma of life. You have wrong DNA, you get wrong messenger RNA, and you get a mutated protein, and that mutated protein can lead to rapid cell growth. There can be activating mutations, there can be regression mutations, and so on. But anyways, the point is the loss in these checkpoints can really inhibit the cellular control, and the cells just keep on dividing without control, and that can be one of the reasons for development of cancer or tumor progression. So again, this is just the same a cell cycle with showing some of these checkpoints, which known tumor mutation genes or tumor-associated genes. For example, you have P53 here, so the gene which is commonly present in prostate and pancreatic cancers. The mutation of these genes is across many tumor types. The P53 is tumor suppressor gene. So once you have mutated P53, the tumor suppression is lost and the tumor goes on. So again, many different, so ATM is a kinase, as a kinase is phosphorylate other proteins and activate proteins. So when you have mutations in these genes, the cell cycle continues, and you can't really stop the cellular growth, and that leads to cancer. And this is one of the major reasons. So cyclin-dependent kinase 4/6, retinoblastoma, E2F, these are again different oncogenes. So most of the oncogenes can function to rapidly grow the cell when the control is lost. So basically, loss in cellular proliferation control leads to rapid proliferation and development of cancer and progression. So again, I think it's really important to remember that cancer, even now, is a major health issue. So 40% of the people are at some risk during their lifetime. So 40 in 100 can develop cancer during their life. At some level of cancer, might not be aggressive, but there's a high possibility or probability that 40 people out of 100 can develop cancer during their lifetime. So the common treatments are radiation and surgery, chemotherapy. I'll go through this. Many of you know what these are. Surgery is just cut the tumor out. Radiation, you expose them to gamma rays. Chemotherapy, you give like a poison. Basically, you give a systemic poison to just kill cancer cell, but you also kill a lot of normal cells, and side effects are very bad. So again, so one of the important things I am interested in, at least the very emerging field, is restoring immune control. So if you think about it, cancer does not belong to the body. So these cells are forming due to mutations in the genome and some changes which are not normal. So if you have a viral infection, bacteria, and any foreign pathogen, the immune system really try to clear these foreign pathogens. So the immune system really looks at cancer as a foreign entity than a normal innate or normal entity. So it produces an immune response against the cancer itself. So we identified that, or many people identified that in 2010, I should say. This field is really new and rapidly growing. So since 2010, a lot of progress has been made to really improve these immune responses. So there's an immune response, but there's also some blocking of these immune responses. So cancer cells are really smart. They try to avoid these immune responses and they grow as much as they can. So these are the emerging hallmarks I'm interested in. This, when I say deregulation of cellular energetics, that what that means is if a cell is under normal condition, it doesn't need a lot of DNA, it doesn't need so much energy. But if it tries to proliferate rapidly, it uses all of its energy to not just to maintain its cellular processes, but also to rapidly replicate its DNA so that it can grow and multiply into different cells. So that happens during cancer. And avoiding immune destruction, so the cancer hides from the immune system by a variety of mechanisms so that the cancer can really grow. So this is what I'm interested in, and this is what we're going to discuss today at a very basic level, although going through some of the interesting therapies. So again, there are other enabling characters like genomic instability, I just said mutations, and there's a lot of inflammation at the tumor area, and sometimes that can help in tumor growth promotion. Next, so again, the evolution of cancer therapy. So very interesting. Surgery is the most used treatment even now. So then comes radiation in 1900s. So then you have chemotherapy in 1940s, where you have chemotherapy given to hematological and solid tumors. When I say solid tumor, such as melanoma, lung, breast tumors. Liquid tumor means heme malignancies such as leukemia, lymphoma, etc. So really, the targeted therapies came in 1980s. These are tyrosine kinase inhibitors, where they really inhibit enzymes or the kinases such as MAP kinase inhibitor, etc. So these really inhibit the phosphorylation of downstream processes or downstream proteins, so the cellular proliferation can be stopped. So then from early 2010, the immune checkpoint inhibitors, or for example, immunotherapies really came. So use of monoclonal antibodies and many different things to really stop the tumor progression. And I'll go through each of these in very detail later. So again, with respect to immuno-oncology itself, the timelines, again, don't try to remember any of these, rather try to listen to this talk as an informative newspaper kind of conversations where you're not stressed to remember, but you might have a pen and paper in hand. If something you really like, you might note that down and do some search later. So again, so this idea of treating cancer using immune responses started in the 1800s. So everybody knows Coley's toxin. What it is, they just take some bacteria and heat and activate the bacteria. So when you say heat and activate, you put it at a temperature of 60 to 70 degrees for an hour, and then you inject this into the tumors, and they see some immune responses. So after that, everything kind of faded off for like almost 100 years later. The IL-2 is a cytokine called interleukin-2, got really working in immune responses. And again, tumor-associated antigens, and I'll go through all these. So first FDA approval of anti-CTLA-4 ipilimumab for melanoma. So this is an immune checkpoint inhibitor. So this relieves the brakes on the immune system and helps the immune system to fight cancers better. So after this got approved, it really picked up speed. And now during the last 10 years, the entire treatment or landscape of cancer has changed tremendously. And scientists who discovered the anti-CTLA-4 and anti-PD-1 mechanisms were awarded with Nobel Prize in 2018. So that's a great exposure for the field itself. So again, starting in the 1800s and really the 21st century, we really see a rapid progression in immunotherapy and the field itself, leading to multiple FDA approvals every year. And as we speak, there are FDA approvals coming out. So again, this is just a slide showing that if you compare the 5-year survival rates across different tumor types, it's a lot of progress between 2007 and 2013, where the checkpoint inhibitors and immunotherapy really started showing up their effects. So if you look at all cancers, the survival 50% to 67% 5-year interval. So that means 50 to 67% of patients were still alive 5 years after treatment across different tumor types. But still, in general, we considered pancreas, liver, lung, and brain as really hard to treat cancers. And there's little progress in these, 2 to 8%, and still a lot of progress we need in these areas to really drive improving survival. So melanoma is one of the best responders for any therapy. Basically, you see 80% to 90% of patients are still alive. So I think in 2020, the survival rate for melanoma is 92%. So if you get melanoma, or if you happen to get melanoma, there are 92% chances that you're going to survive for the next 5 years. So it doesn't mean that you get melanoma, but it's comforting to say that the treatment landscape has changed so much that you're almost 90% of the people are still alive after 5 years of melanoma. So again, that's just a schematic showing that how we are constantly improving the survival benefit of the patients with all emerging therapies. So just to give a brief understanding of what's the difference between the traditional treatments and cancer immunotherapies, as I just mentioned. So as you all know, chemotherapy, surgery, or radiation, I consider as the traditional treatments. The treatment basically kills the cancer cells. At the same time, it's not very specific. It also kills the healthy cells where you have gross destruction of healthy tissues as well. So, and the side effects, if you look at the side effects, it's pretty alarming. And post-treatment protection. So what do I mean by post-treatment protection is recurrence of the disease. So you have a tumor, for example, in your lung, you take it off, you do a surgery, and again, you recur in 6 to 8 months. So that doesn't help you. Basically, it's a temporary control of the tumor. At the same time, it's a very risky treatment approach where the patient, you lose white blood cells, you lose many different important lymphocytes, and really it's not good for the patient. And still, there's no other options until 2010, and patients used to go on these treatments. So with the new cancer immunotherapy, so it really uses our immune system, or the patient's immune system, to specifically target cancer cells while leaving the healthy cells. The side effects, obviously, there are side effects, but they are minimal, and the post-treatment protection is very high. So if you think about immune responses, not just for cancer, for any different pathogen or viruses, so you have a resting T cell, then you get into activation where the T cell grows, and then you go into a stable period where the T cell doesn't proliferate, but they are stable. Then they go into resting, where they go, these responses calm down and they go into memory. So there is evolution of responses. There's a plateau phase, and they go to memory.
9 o'clock.
But then the whole point is, once they go into memory, if they re-see that, or if they reintroduce this antigen again, the memory T cells rapidly proliferate and provide immunity. So that's memory responses, and that's really important for constantly controlling the disease or constantly controlling the cancer in these treatment modalities. So again, many of you might already know this, but I think for the purpose of this talk, it's important to understand the type of immune responses. So there's innate immunity, where that's the first response. Again, with cancer, we don't deal with innate immunity as much as with pathogenic responses. So many different cell types, as you already know. The adaptive immunity is what is most important for the cancer purposes. So in the adaptive immunity, you have B cells secreting antibodies. So plasma cells are the B cells that secrete antibodies. And the T cells, they can directly kill a target cell. So this is called cell-mediated immunity. And mainly for the purpose of cancer, most of the research, or most of the understanding, is that the T cells are really important, as they can directly target any given tumor type. So particularly the CD8 T cells are the most effective, or we call them cytotoxic T cells, as you know, and they can kill the tumor cells directly in the context of MHC class 1, major histocompatibility class 1, also called HLA, human leukocyte antigen, for the purpose of humans. Again, so what happens, again, don't get confused or don't be scared by these images. Think about it, it's a very simplistic view. So any cancer or any cell undergoes apoptosis and releases tumor antigens. So in case a cancer cell die due to due to chemotherapy or radiation or due to a tyrosine kinase inhibitor, so then you have these tumor antigens. So when I say antigen, these are short peptides. So these are mutated peptides. So these are a result of the tumor. And these can be really uptaken by dendritic cells. These are antigen presenting cells. So they can really take endocytosis, these peptides, and process these peptides. So they have to cleave these peptides. So for MHC class 1 purposes, they have to cleave them for 15 amino acids. For MHC class 2 purposes, it's a little short, 8 to 12 peptides, I believe. Anyways, they present these to T cells in the lymphoid tissues, a draining lymph node, etc. Then this is called activation and priming phase. Once the T cells are activated, they go into the bloodstream, and again, they find the target cells that in the context of MHC class 1 for CD8 T cells, MHC class 2 for CD4 T cells, and they release cytolytic enzymes and kill the tumor cells. So they really kill the tumor cells in the context. So I think I have to show this video, which is really nice. So this is showing a T cell directly killing a cancer cell. So the green cell is a T cell killing the red cancer cell. So anyways, that's how the T cells, they directly attack and kill the tumor cells by releasing some enzymes, which really, these are called cytolytic enzymes, for example, granzyme and perforin. They really kill the tumor cell. They basically put holes in the tumor cells and they destroy tumor cells. I'm trying to switch my slide. Next slide, please. So in the context of a T cell killing a tumor cell or activation of T cells, so there are several signals for a T cell to become active. I think this is really important. So what is present on a T cell? So for example, in the context of a CD4 T cell, you have the T cell receptor binding to a MHC class 2 of an antigen presenting cell, such as a dendritic cell. So this is MHC class 2. You can really tell because it has 2 alpha and 2 beta chains. The MHC class 1 has only 2 alpha and 1 beta chain. So that's how you differentiate class 1 versus class 2. So this is the first signal. So then you have the CD28 binding to B71. So these are called co-stimulatory signals. And this really causes survival in T cells and rapid proliferation. Then the antigen presenting cell or dendritic cell also releases cytokines that basically acts as fuel to the T cells and keeps the T cell going on. So you have 3 signals. One is the first signal is the TCR binding to MHC, then co-stimulatory signal, and then the cytokines are released. So if there is only one signal, for example, if you only get a signal 1 and there's no signal 2, then the T cell is not fully activated and they're not functional. They're not fully functional. So again, if you activate these pathways using antibodies or using any different small molecules, you can really drive T cell function. Or you can give a cytokine therapy to a patient, you can really improve their T cell function. So these are many different ways how the T cells get activated. And using this understanding, I will go through the talk where each of these pathways can be activated to really see enhanced immune responses for the treatment of cancer. Again, so between antigen presenting and T cell, there are many different signals. Okay, these are not well studied, or these are constantly being studied. Some are activation signals. So these are called co-stimulatory signals. So this is just giving an extra push. Think about you're trying to lift something very heavy, and you have your friend coming and helping you, giving you another hand, and that really helps you to complete your job. So these are that signals. which can really help improve the responses. So you call them activation signals or co-stimulatory signals. So in another context, so if you activate these, you have more T cell function and you drive more immune responses. On the other hand, you have inhibitory signals. The red rectangle highlights these. So inhibitory responses are they inhibit these T cell activation, or they inhibit the T cell activity. So binding of PD-1 to PD-L1. So PD-1L1 is a ligand, PD-1 receptor, can really inhibit the T cell function and drive the T cell into apoptosis and suppression of immune responses. Then you have these B7-1, which has to bind to CD28. CTLA4 competes for this binding and these, again, inhibitory signals. So if you block these inhibitory signals, again, you can activate T cell responses. So either you can activate stimulatory signals or you can block inhibitory signals. So again, the first Nobel Prize in immunotherapy went to the scientists who really identified these pathways, CTLA4 and PD-1. PD-1 stands for programmed death pathway. CTLA4 stands for cytotoxic T lymphocyte antigen pathway. So again, everything is like very emerging. This is very new. So research is ongoing at every given molecule. So, but the PD-1 and CTLA4 are the most important, and there are many FDA approvals across different tumor types. So moving to that. So I'm talking too much, and I think I should really tell you that does immunotherapy work, and show us some evidence that this works. So again, I think many of you might not be very used to seeing this kind of data. So this is called the Kaplan-Meier curve. So what this really shows is the blue line indicates the patients treated with dacarbazine, it's chemotherapy. So if you see a drop, like if you see a drop at any given location, that means the patient have died. So again, this is showing the patients without progression, percentage patients without progression at given months. So these patients, for example, did not progress at 12 months, and they're 20% or 10% given the chemotherapy. But again, if you look at nivolumab, which is anti-PD-1 antibody, which I just told you about, so anti-PD-1 antibody blocks the inhibitory pathways and really activates immune responses. So 40% of the patients are still progression-free at 15 months. If you look at the actual numbers, so 108 out of 210 patients were who died or had disease progression. So you have only 50%, but here 163 patients have died or had disease progression. So it's really, really tremendous benefit to the patients. Again, as a single agent, the median survival, so every patient lived 5 months without a disease in the immunotherapy group compared to 2 months in the carbazine. So again, a 3 months extension in survival for cancer patient is considered a tremendous increase in survival with nivolumab, again, which is very less toxic than chemotherapy. So again, this really works. If you look at the curves, they're really separated. And these patients, these set of patients continue to live without progression. So this tells us that immunotherapy really works and the patients really respond. But again, it's important to understand that not every patient responds, and there are some patients who don't respond, and we'll go through why they don't respond and how can you make these patients respond, and really by combining different immunotherapies and understanding the resistance mechanisms of why these patients don't respond to monotherapy or a given therapy. So there are many different classes of immunotherapies. I think I'm not going to go through all of these, but I'll go through the most important ones and give some level of understanding on how these work. And again, you can take notes and really look this up after. I did mention the checkpoint inhibitors. I think the Nobel Prize was given in 2018. It's not 2019. But again, these are the really these are the therapies which really change the landscape of treatment. Then you have CAR T cells, and you have vaccines, cytokines, and oncolytic viruses, which I really work with. And then again, for this slide, it considers metabolic inhibitors and chemo and radiation as kind of immunotherapies because they do activate some level of immunotherapy, immune responses. But again, I'm not going to go through this part today. I'm just going to go through the most interesting or the most emerging immunotherapy. So the first one I would like to go through is immune checkpoint inhibitors. So again, there's a short video. I believe, can you see the video right previously? Dr. Ramchandran and Dr. Sumalata, did you see the video last time?
No, actually, we could not see the video.
Okay, all right. So then what I will do is I'll just share my screen so you can see that, you can see the video. I believe you can see it now, right?
Yeah, you can see.
Our immune cells protect us from harm, patrolling our blood in search of potential problems, and travelling to sites of infection or damage. These cells need to spot the difference between healthy and unhealthy tissues. For example, when our cells are infected with a virus, they need to be destroyed. Infected cells display different molecules on their surface from healthy cells, indicating they're damaged. Immune cells can recognize these signs of disease and pass on instructions that tell the infected cell to self-destruct. But when it comes to cancer, the situation is a lot more complicated. For a tumor to grow, it needs to avoid detection. One way it can do this to produce molecules that can trick the immune system. One of these is called PD-L1. It interacts with a molecule on the immune cell, telling it to ignore the cancer. But researchers have now developed drugs to interfere with this process, allowing the immune system to attack the cancer. And for some cancers, these new immunotherapies look set to change the way the disease is treated.
Again, that's how these antibodies work. They just basically block the pathways and to improve the immune responses. And that's why they're called immune checkpoint blockers or immune checkpoint inhibitors. All right, I'm trying to slide. So again, so if you see the slide, without immunotherapy or without immune checkpoint blockade, the PD-L1 binds to the PD-1 and suppresses the T cell activation or inactivation of T cell, and the tumor really escapes. But then with immunotherapy or with anti-PD-1 antibody, this interaction has been blocked and the activation of T cell and constantly eliminate the tumor cells. So there are two different checkpoints. It's called CTLA-4, cytotoxic T lymphocyte antigen, and the programmed death ligand 1 and receptor. So that's what I described here. So again, so in general, the body uses these checkpoints to avoid autoimmunity. So for example, if you think about this for a second, so in organs like your lungs, your gut, your stomach has a lot of bacteria to keep homeostasis. Again, and again, same thing like your lung. When you breathe in, you have a lot of bacteria, etc. So these checkpoints avoid the immune responses at that areas, and again, to really inhibit the immune responses and autoimmunity. So cancer tries to take advantage of this so that it can grow. And so blocking these immune responses can be really beneficial. So the difference between CTLA-4 and PD-1, again, is so the CTLA-4, there are two phases, as I just mentioned. First phase is a priming phase, where T cell has to get activated. So this happens really in the lymph nodes or lymphoid organs. So the CTLA-4 just inhibits the priming phase of T cell activation. Okay, so the CD28 binding to B7-1, as I just described, is inhibited by the CTLA-4, and then you can block this by using a CTLA-4 antibody, for example, ipilimumab. The effector phase is where it happens at the tumor site, where the T cells really kill the tumors, and because of PD-1 and PD-L1 interaction, this is reduced, or this is the tumor cell just escapes these immune responses, and you can block this using PD-1 or PD-L1 inhibitors, for example, nivolumab. So just think about this. There are two different checkpoints in the immune responses. One at the activation stages or the priming phases, it's called CTLA-4. The other at the effector phases, it's called PD-L1. So blocking these inhibitory pathways can really help patient survival and generate tumor responses. So this is a PET CT scan of a patient. So this is pre-treatment. So what they do, I mean, you can read upon PET CT scan. They inject glucose with a dye, and then it localizes to areas of high metabolism. For example, you see it's very dark in the brain. That means it's a lot of metabolism going on. But then you overlay this with a CT scan, and you really find these spots of tumors. So I think this patient have lymphoma. So you see these dark spots of places you're not supposed to see. Again, at the lymph node, probably. And then after chemotherapy, you got some response, but again, these internal disease and everything still stay on. But after giving immunotherapy, look how clean is this area. You don't see any of these lymphoid or these leukemias, or you see any of these cancers. And again, so not just in this area, you also look at the head, the neck region. So that's just blood. So again, this really tells us that it's superior to chemotherapy and works, and really beneficial to the patients. So again, this is the survival curve showing 12-month survival post treatment work published in New England Journal again in 2019, showing that ipilimumab is CTLA-4. You get 37% of the patients respond or who survived 4 years after, and then 56% with nivolumab, which is PD-1, but the combination is superior, showing 70% almost responses or survival at 48 months post-treatment. So again, these are really tremendous responses in melanoma, showing that immunotherapy really works. And that's why these immune checkpoints are really change how we treat cancer today. And that's one of the reasons I believe these scientists were awarded with Nobel Prize. So again, as I just mentioned, 20 to 30% responses before immune checkpoints, ICB'S or immune checkpoint inhibitors, which now is 70+% with combination immunotherapy. So again, there are some common side effects like pneumonitis, nephritis, colitis, where when T cells become too active, they go and kind of attack the cells in the lung, in the kidney, and in the gut. Again, you can use steroids or some antibodies to block these responses, and the toxicity profile is not too bad compared to chemotherapy. I think if you take anything from this talk, understanding what's immune checkpoint inhibitors and how they change cancer treatment, this is the most important thing. And again, leading to almost 30 FDA approvals in different cancers during the last 5-6 years is tremendous advancement in the field. So adaptive cell therapy. So basically, what happens these are different therapies. So in these, you really introduce cells back into the body. So there are two different types of adaptive cell therapies. I think some of you might heard about CAR T cells, chimeric antigen receptor T cells. The other one is TCR-T cells, so the TCR is modified. So again, I don't want to go into too much details, but again, to give a brief flavor of how these work. So you obtain a patient's T cells. So if a patient walks into the clinic, they'll get their T cells. They'll insert a gene in their T cell by genetic engineering. And then this can be a modified T cell expressing a chimeric antigen receptor, which I'll discuss in a second, or a T cell receptor. Then these are proliferated in a dish. So these are grown in a dish, and then they're injected back to the patient. So these cells can now detect tumors and kill the tumors. So approximately they inject back 1 billion cells. So as I just mentioned, the MHC and the TCR interaction, for example, if you are trying to target a B cell malignancy or B cell lymphoma, you generate a CAR against CD20 because B cells express CD20. So these CAR T cells go and bind to CD20 and destroy the B cell malignancy. So these are really precise. So this is a very laborious process where you take the T cells out of a patient's body, then engineer, grow these T cells. Again, this is all done in GLP, GXP environment. There's a lot of investment going on to this. And again, this really changed the landscape of heme malignancies or liquid tumors. So the immunotherapy, immune checkpoint blocker really changed the landscape of how these work in solid tumors. The CAR T cells really changed the landscape in liquid tumors, the hematological malignancies. So there are two different CAR T cell therapy approaches I just mentioned. One is called TCR-T cell The other one is a CAR T cell. So they're two different things. So this is your endogenous TCR. So this is your T cell receptor, which is present normally. In a transgenic TCR, you just replace the alpha beta, V alpha, V beta chains of the T cell receptor so that it can target a specific antigen. So you take the T cells, you express these genetically modified T cell, and you put them back. So again, here it's still MHC dependent. So if you take a patient T cell and you can't give to another patient because these are MHC or HLA dependent. In this CAR T approach, what you basically do is you take an antibody variable light and heavy chains. So you join them with a linker, and then you insert different co-stimulatory molecules into these linkers, and you produce a CAR T cell where it's a chimera. So a CAR T cell is called a chimeric antigen receptor because it has the parts of both an antibody and a T cell. So B cell secrete antibody. So you basically use the advantage of the variable regions of light and heavy chains to be engineered on a T cell. So if you think about this doesn't need an MHC and can recognize only surface proteins. But in the TCR, it can recognize both surface and internal proteins, and it needs MHC, and it's MHC dependent. So just for your knowledge, think about there are two different approaches in T cell engineering. One is chimeric antigen receptor T cell, or CARs. The other one is called TCR, transgenic T cells. As I just mentioned, in CARs, they take the single chain fragment variable. So that just means the variable regions of the fragment antigen binding the FAB part, and they link them with the linker. And then based on the generation of CARs, they also link them with co-stimulatory molecules. And if you might know the molecular biology, these are genetically transduced into a CAR, into a T cell, where the T cell will now express the TCR as well as the CAR, chimeric antigen receptor. So CAR T cell can attack the tumor irrespective of their HLA, because this is a FAB of an antibody which doesn't need a HLA, which can just recognize proteins or antigens on the surface. So again, I think I've mentioned this already. TCR is different. So this is CD8 T cell binding to MHC class 1, has only one beta T chain. Again, you replace this T cell receptor with a tumor-specific T cell receptor. With the CAR T cell, you replace the T cell receptor with a B cell specific antibody. So basically, you take the single chain fragment variable, the FAB portion of an antibody, and then you engineer on the T cell so it can recognize independently. So again, these are some comparisons. So problem of mispairing with endogenous TCR. So you try to express TCR, you might not change everything. Here you don't have that problem because this is not a T cell receptor. It's a completely different entity. And really, you can track these in an animal model. So this is an IVIS imaging showing the CAR T cell proliferation. So this is bioluminescence. So the T cell is engineered so that it expresses firefly luciferase. Then you inject these T cells into a mouse model where you on day 1 you see the presence here, but day 6 and day 13 they really go around and you see they go and attack the cancers that present in different areas. So it just means the CAR T cells can really proliferate in animal models or humans and attack tumor cells. So both this is really important. So again, this is really important to know that the first patient treated with CAR T died in 2018. So it's very sad that after infusion, the patient died in 3 months before the next therapy. So later, the patients are treated with steroids or anti-IL-6, tocilizumab. It's an anti-inflammatory cytokine, or it's an antibody which can block the inflammatory responses. So the cytokine storm is really calmed down, and the patient. So think about it. If you're infusing a lot of cells into human body, you create a lot of cytokine storm, and this can be really blocked using the IL-6 antibody. So it's important to mention that in 2012, the first patient treated with CAR T really responded. Her name is Emily Whitehead. 6 months in 2012, she had a complete response, and even today, after I think 8 years, she's still cancer-free. And this is one of the reasons where the CAR T-cell really kicked off, and during President Obama time period, they had precision CAR T and really drove a lot of funding into this research. And again, before CAR T, the survival rates in lymphomas is 20 to 30%. Now it's 70%. So a tremendous improvement in survival for patients. So if anything, do you remember about heme malignancies, leukemia, lymphoma, myeloma, etc.? The CAR T cells have really done a tremendous job in improving survival for patients. So the next thing is tumor vaccines. So many of you might heard about vaccines in general. So what does a vaccine do? Think about it. Excuse me. If you're getting a vaccine for a virus, for a flu or for corona, what you basically do is you vaccinate or you inject heat inactivated proteins or live proteins or live some sort of inactivated antigens from the virus to create an immune response. So same thing in cancer. What happens is the cancer vaccine idea originally started when you have these mutated genes. So the genes are mutated, leads to mutated RNA and then mutated proteins. So you identify these multiple DNA mutations, which can lead to tumor formation. So mutations can develop new proteins. These are called neoantigens or tumor antigens. So the term neoantigen just means in normal conditions, these antigens, so antigen is just a protein, which is derived from mRNA, which ultimately came from the DNA sequence. So is not present in normal body, but due to mutations, you develop these new antigens. So these may lead to tumor formation. Taking this into consideration, so you have a mutated peptide in a tumor. So this is mutated antigen. Again, as I said, it goes into the DNA and it goes into the RNA, and you form a mutated protein, which is taken up by the proteasome and cleaved and then presented in the context of HLA, and then the T cell responses are created. So taking this idea into consideration, a lot of efforts have been driven into tumor antigen discovery. So again, this is so what they do is take a tumor cell or take a bulk of tumor, then they identify the DNA sequence. So they really identify the mutations in the sequence. When they identify these mutated sequences, they generate mutated peptides. So again, all mutated peptides are not immunogenic. So they really identify which mutated peptides are immunogenic by really exposing them to T cells. And for example, let's consider that this peptide red has grown a lot of T cells. So this can be a candidate for a vaccination. So they have a lot of prediction algorithms, etc. To do this, again, this is very laborious. It can be very expensive. But again, these are rapidly emerging technologies where you can save someone's life. Sometimes no matter how much money you spend, you can't save someone's life. And putting that into context, and giving these kind of therapies can really save patient lives. So then you identify particular antigens or a pool of antigens, and then you vaccinate. You basically inject that into a patient with an adjuvant. So this is just a vaccination approach where the immune responses are created against these mutated peptides and finally lead to tumor clearance. So basically, you take the tumor, you sequence it, you identify these mutated proteins, and see which of them are mostly immunogenic. Once you identify these immunogenic peptides, you vaccinate, and then that creates an immune response. Again, these are still pretty new and pretty advanced techniques. I understand there are logistical difficulties, but again, companies are doing this. They're spending a lot of money trying to do this and really working in the context of patients. So the cancer vaccines can be many different types. These can be cell-based vaccines, where you take antigen-presenting cells, for example, dendritic cells from patient, and you stimulate them with peptides and inject them back into the patient. Or peptide vaccines, which I just said, you can just inject the peptide vaccine with an adjuvant. Or viral vectors, so you can really give viral vaccines. Like if you give the viral vector, it releases the functional protein and produces immune responses. Again, gene-based therapies, where you can give RNA vaccine or DNA vaccine. So anything encoding a tumor antigen and produce a tumor-specific response is just a tumor vaccination strategy. The final goal is to mount or produce an anti-tumor specific immune response, which will eventually lead to the tumor clearance. So that's all with the cancer vaccine. So everything I mentioned until now is different in the case that they act at a T cell level, but they cannot drive T cells into the tumor. So oncolytic viruses are something very unique, which I work on, and they can really bring the T cells into the tumor. So again, if you inject this into a patient, there are mechanisms that differentiate healthy cells versus normal cells. So the healthy cells, they don't replicate the virus and they're not damaged. The normal, the tumor cells, they're susceptible to the virus and they die. And the virus replicates and the tumor cells are destroyed. So once, upon destroying, the tumor antigens or these tumor peptides are released, and they also produce a systemic anti-tumor immune response. So it's a one-shot two bird kind of situation. It can directly kill the tumor cell, and it can also activate immune responses. So again, the first step is selective. So the patient been injected with the virus. So T-VEC, Telmagene, Leperhavic is the only FDA-approved oncolytic virus based off herpes simplex 1 for the treatment of melanoma currently. The healthy cells does not replicate the virus. The cancer cells replicate the virus. And again, you can engineer these viruses to express different cytokines, costim molecules, etc. So the tumor cells die due to direct lysis effect. Then comes GM-CSF releases in the tumor microenvironment, and you can really produce an immune response. And again, death of not just the infected tumor, but also the distant cancer cells. So this is a patient showing that local injection can cause a systemic immune response. So in this patient, this lesion has been injected. So this is a pre-treatment. This is 3 weeks after treatment. So you see a loss of this, the tumor goes in necrosis, and the other tumors as well disappear. So 6 months and 9 months. So as you see this, not only the injected, but also you see activity on the uninjected tumors. It's really important, showing that a local injection can cause a systemic anti-tumor immune response, leading to tumor clearance. So again, I've investigated these mechanisms in mouse models, showing that when you combine the virus with no tyrosine kinase inhibitors, it really improves survival. Again, this is Kaplan-Meier curve, showing that anytime you see a drop in the line, that means an animal died. So you have some complete responders given with the combination of the virus and the tyrosine kinase inhibitors. So at 100 days post treatment, a set of mice, at least 40% of mice, still survive and continue to survive. Later, what we did is we re-challenged these mice. So, for example, if we take a mouse and the primary tumor is completely gone, then we re-challenge or re-implant the cancer cells into these animals, and show that most of these animals are immune to re-challenge. So you give them cancer and they don't even grow. So this tells that there's an immunological response and there's a memory response due to the treatment. So the memory component is really important for the immune system to really have the memory, so that when you get this cancer back, it can really act and produce tumor control. So again, if you know, this is a very important slide. So this is a flow plot. When you do flow cytometry, you make these plots. So what this is basically showing is these are different treatment approaches, showing the percentage of antigen-specific T cells. So if you have T cells, you can have a T cell specific to many different antigens. What we ask here is, can the treatment produce antigen-specific T cell responses? If you don't have antigen-specific T cell responses, it's basically useless. So GP100 is a melanoma antigen, and tyrosinase is also a melanoma antigen. So with the treatment, the combination treatment, you see 12% of the T cells are specific to that particular antigen. So that means these are not T cells just hanging out there. These are T cells very specific to that tumor antigen. They're rapidly killing the tumor. So that's really important to show that the responses are specific to the tumor type. So again, this is a gene expression profiling, showing that increase in gene signatures during treatment. So most commonly upregulated genes are associated with T cell activation, etc. So again, this is called a heat map. So if you have, for example, 100 patients where you analyze 100 genes, instead of showing on an Excel sheet, you show on a heat map where the blue indicates lower levels of expression and the white indicates medial expression and the red is higher levels of expression. So increase in these gene expressions really are leading to tumor inflammation. And T cell activation. So that's really important. And again, just to end this, you have 3 different animals treated with the virus. So again, this is really replicating that scenario in the clinic where you have a primary tumor and you also have a metastatic tumor. So you don't treat the metastatic tumor, but you only really treat the primary tumor. So in some cases with the partial responders, where you see the primary tumor completely go away, but you still see the metastatic, although there's tumor control. And in complete responders, you really see both these tumors go away. So again, this tells us not every patient responds and not every animal responds. So they really need to understand the mechanisms, how each of these therapies work, and really convert these patients to respond. Again, this is immunohistochemistry showing different proteins at a tumor level. So the CD8 T cells are stained by green. So at week 6 after treatment, you see a lot of green. At week 30, you see a lot of green. And again, you see blue is staining the melanoma. As you grow into week 30, you see the reduction in blue stain. That means reduction in melanoma and an increase in immune cells. So they're really showing that the viruses can really bring in T cells and reduce the tumor in patients as well. So again, this is the final conclusion of the work, showing that when you have tumors that does not respond to immunotherapy, or does not respond to checkpoint immunotherapy, you can convert them using the virus to more checkpoint responding, and you can lead to 100% tumor clearance. So this has been published multiple times, and we have a patent on this, really showing how these tumors can really respond, how the viruses can really make the tumors which does not respond to traditional immunotherapy to improve responses in patients. So the next one, I don't want to go too much into the cytokine therapies, which many of you might be aware. So these are just cytokines. So many of you know that the T cells or the other cells produce cytokines. So one thing to understand is in cancer immunotherapy, most of these cytokines are called gamma chain cytokines. So they have a common gamma chain, so which is really important, and they act at a T cell level to produce the immune responses. So the cytokines basically act as fuel to cells. Most of them signal through gamma chain, and then you can have unwanted side effects. So you have too much cytokines. They also bind to the alpha chain and beta chain, for example, IL-2, and produce unwanted side effects as well. So you can look up all these, but just to let you know, also cytokines can be injected into the tumors or injected systemically to produce or to improve immune responses. So I'll talk about one cytokine which is really interesting, and how, because this is the first approved immunotherapy. So IL-2 is interleukin-2. This is one of the first approved immunotherapy. IL-2 helps in CD8 T cell proliferation. That's very good. But IL-2 also helps in the growth of inhibitory cells. So these are called regulatory T cells. They release IL-10, interleukin-10, and really suppress the T cell responses. So what people are doing these days are they're creating biased forms. So, for example, you have a normal T cell, and IL-2 sticks onto this T cell. This is the inhibitory cell population. It binds to alpha, beta, and gamma, and really increases or suppresses the immune responses in this case. When you have a biased form, so for example, in this IL-2, you modify the IL-2 so that it can only bind to alpha and beta, it can really fight cancer, and it can really do effector T cell functioning. So how they do this is they take the extracellular domain of IL-2 receptor alpha and fuse it to IL-2 cytokine and produce a molecule which can specifically bind to the CD8 T cells and alpha beta gamma chains, so that the inhibitor responses are lost. So these are fusion proteins. There's linkers between these proteins. So basically what I'm trying to say here is, even the cytokines not working so well, the new advancements in cytokine research is they're modifying the cytokine in such a way that it can only do the beneficial effects, rather masking the inhibitory effects. So the other forms of cytokines they're doing are pegylated forms as well, where they pegylate the cytokine, increases the half-life, or they bind the cytokine to an Fc portion of an antibody. So anything you can think of to do to conjugate this cytokine with anything possible, it's still going on. So the last but one I want to discuss, again, you can write this down and really read up if you like, but I'm just giving you what is available. These are called BITEs, or bispecific T cell engagers. So what they do here is, so you have two different antibodies. For example, one is detecting the tumor, the other is directed to CD3. They take off the single chain of the heavy chains of the two antibodies with the link. So it's called a bispecific because two different groups together. For example, if you have a magnet in one hand that can specifically bind to the iron, and if you have another magnet in the other hand that can specifically bind to gold or platinum, so if you're considered as a BITE because you're engaging two different things. So think about here. So you have a tumor cell expressing tumor antigen. So this portion of the bispecific has an anti-tumor specific antigen single chain fragment variable. The other side has C3, and so the BITE really brings them together and these hey, kill the cancer. These are not very approaches because this is one fit for all purposes. Because BITEs can be developed for many different. For example, these are off-the-shelf, easy manufacturing, unlike the CAR T cell. For example, BCMA BITE for myeloma. So these are two ends. Okay, this end binds the cancer cell, and this end binds the T cell, and really bring them together so that they can really the T cells can kill the tumor cells. There are many different MUC16 or MUC17 is for PSMA is so these are different bispecific which are currently in development across different tumor types. So what they basically do is they physically marry a T cell to a tumor cell and helps the T cell to kill the tumor cell. So the last one on my list is antibody drug conjugates, which is really interesting. If you think about chemotherapy, chemotherapy works, but it's not specific. It kills all the tumor cell. So how can you target any drug, not the chemotherapy or tyrosine kinase inhibitor or any small molecule? Think about this. If you know molecular biology very well, anything you can code with the DNA can be genetically encoded. But small molecule, we talk about therapy. The chemistry that. So if you want to specifically deliver these to a given target cell, what you do is you find a receptor expressed by a cancer cell and you develop an antibody against that, and then you conjugate this antibody to a linker with a drug, so that when these antibody drug conjugates bind to the cancer cells, they're endocytosed and then releases the chemotherapy and kill the tumor cell. These are very specifically delivering. These are delivering the cargo to the very specific tumor cell. So, for example, if you want to deliver a letter to a person and there are a million persons, you specifically detect that person by really giving a specific antibody to the target receptor, and then gets internalized. So the conclusion of this talk, again, I don't want to inundate you with a lot of these therapies, but I think the idea here is if I can go through each of these are very important therapies. Again, there are many which I did not discuss today. Please give me a flavor of the kind of therapies out there so that we can really think beyond what's existing chemotherapy or surgery or immune checkpoint inhibitors. So the goal here is if you look at survival at 60 months or 96 months, chemotherapy and targeted therapy, you see very poor, almost 10% patients are alive at 60 months. It's very less. Ipilimumab is a CTLA-4 and PD-1 blockade. You see increase in response 30%. But the combination of these two immune checkpoints really improve their responses to 70%. And again, with further identifying in other combinations, improve these responses further. So it's hard to reach the 100% mark, right? So we're understanding each of these therapies so that what can you combine in a given patient and really develop rational combination approaches for patient survival. Again, I'm really sorry if I went over a little bit, but again, I want to inform the group that different therapies are existed, and then you can really understand. So again, thanks to everyone. for inviting me. And I work with many super smart science people and doctors at Harvard Medical School, and collaborate with Amgen as well. Particular thanks to the organizer. Thanks to everyone. So this is my PhD committee. I really like to show pictures. So this is Dr. Bertino. He's the legend in chemotherapy. He identified the mechanisms of methotrexate resistance. And again, these are tremendously smart people. I'm really happy that I get to know them and work with them to really know what I know today. I'm very thankful. So this is our lab at Harvard. And again, all these people are super smart, very energetic. Again, this is lab at Sloan Kettering. I'll take any questions. So one thing I want to conclude is, so it's been almost 16 years since I started GIET School of Pharmacy, and almost 10 plus years, 11 years since I moved to the US. And everything I've learned is really important at every different phase of your education. Sometimes you think, I'm studying biology, I don't need to know math. It doesn't work like that, unfortunately. If you want to be good at something, you have to be good at many things as well, so that you have the proper background to really understand. So immunology people have to understand molecular biology, pharmacology, everything. You think this as a subject in science, and really understand science rather than a subject. Try not to remember things. Rather, if you understand something, you kind of remember it at a very deep level. So with that, I really thank everyone. I'm sorry, a little over, but I'm happy to take any questions now.
Dr. Praveen, not only your team, you are also very smart. You people are really great in cancer research. It's really an impressive presentation on cancer immunotherapy, Dr. Praveen. You hear my voice, Dr. Praveen?
Yes, I can hear you.
Yes, I'm sure that all the participants must have benefited with the updated information you gave on cancer immunotherapy. Now we have some questions from the participants. Shall I present the questions to you?
Sure.
There is one participant. There is a question from one participant. What are the ways to reduce side effects of radiotherapy in cancer treatment? So what are the ways to reduce the side effects of radiotherapy in cancer treatment?
So again, I'm not a radiation oncologist. That's a very specific question to a radiation oncologist. But I've worked with many people in my experience. So what they use right now is called a gamma knife. So previously, the radiation is given. It's called grade. So there's different grades of radiation. Grade is just a grade, GRA, of radiation. So with the gamma knife technology and the emerging radiation, so they mix the beta and gamma rays and specifically target that area so that it's very specific. And then the gray of the radiation is also very customized for that patient. So the radiation itself is very complicated. So if you ever go into the radiation room, which I did, so you have a patient lying on the bed, you have 6 different rooms around that radiation therapy. So one room has a physicist. These physicists control how much of the nuclear energy has to come at that time. So if you think you have a tumor in your lung and you're giving radiation, every time you breathe, the position changes. So they also take into consideration your breathing patterns, your movements, so that the radiation is very precisely targeting the tumor. So there's a lot of effort into this. And again, if you're interested, look at the gamma knife. That's the more precision radiation these days. But again, I'm not qualified to give the exact answer because I'm not a radiation oncologist. But I can speak to that level.
Thank you, Dr. Praveen. There is a next question from another participant. Whether immunotherapy lead to any autoimmune complications?
Exactly, that's true. So I did mention in the immune checkpoint blockade. So at every given tissue, so for example, the lungs, the gut, or the stomach, or the kidneys, where you have contact from a lot of external objects, you have your immune responses constantly slow down on purpose so that the T cells does not attack your lungs because they have a lot of bacteria. You breathe in bacteria. So in that cases, you have side effects, autoimmune side effects. These are called, for example, pneumonitis, nephritis, gastritis, where the T cells of your own body target your organs. But again, as I said, you can use steroids, dexamethasone or prednisone can be given to really calm down these effects. And again, That's a common side effect. So one thing everybody have to understand is you have to balance the benefits versus the side effects. So if you're a healthy person, you are obviously worried about autoimmunity. But if you're a cancer patient dying, so that's not a major concern. But again, there are some toxicities, and that there can be managed.
Thank you, Dr. Praveen. There is another question from participant. Is there any effective immunotherapy for lung cancer? As the survival rate of lung cancer patients are very less compared to other cancers.
So lung is a totally different game. So lung cancer again is divided into many different things. I mean, my specialization is melanoma, but lung is small cell and non-small cell lung cancer. So PD-1, pembrolizumab, has been recently approved for lung cancer. And again, there are studies using viruses in combination with checkpoint blockade in lung cancer. So so I think the important thing to understand there is you have gross inflammation in lungs because of smoking or genetic disturbances, where it's hard for the T cell to penetrate into the lungs. And one thing they're doing right now is, so commonly what they do with immunotherapy is an adjuvant setting. What do I mean by that is you give surgery first and then you give immunotherapy. With lung and other hard to treat tumors, they're going into neoadjuvant approaches where they give the immunotherapy first, then remove the tumor, and again continue giving the immunotherapy. So these are some advances with lung and other hard to treat tumors. So again, many problems, constant research, solutions are awaited. That's the answer for that.
Thank you. Very good, Dr. Praveen. There is the last question from the participant. Is this therapy suitable for, I mean, immunotherapy suitable for all age groups? Is there any resistance during therapy?
No, so far there's no evidence that age is a matter. Obviously, when you get very old, I mean, in our clinical trials, we are treating 95-year-olds and we see responses. So again, at every level, you have some kind of immune responses in your body. And again, the dose might be different for neonates or pediatric patients compared to adults based on the kilogram body weight and the immune responses. But so far, there's no evidence that it works in a different age group and sex or any differences. So basically, no differences so far.
Thank you, Dr. Praveen. You gave very convincing answer for all the participants. So now I hand over the session to Dr. Sumalata for vote of thanks.
Yeah, can I say something? I really thank giving me this opportunity. I've known everybody since a long time. It's a pleasure speaking here. So someone has a really important question. I look in the chat. So can you now say immunotherapy can be considered as a first line of treatment for cancer? Absolutely, yes, for melanoma and many cancers. If you have a melanoma, you go to this cancer hospital, they first treat you with checkpoint blockade. So many tumors, it's changing. It's not surgery, but immunotherapy. Thanks. Sorry.
It's okay, fine. Thank you very much, Dr. Praveen. It's a great privilege to me to extend my vote of thanks for this fabulous session. Firstly, I thank the protagonists of PharmaFit, Dr. T.V. Narayana sir, the President of Indian Pharmaceutical Association, for the strenuous efforts to upgrade the profession of pharmacy. I owe my special thanks to this distinguished speaker, Dr. Praveen, the profound researcher in oncology. Really, the very presence of this speaker, in spite of his busy schedule, is a reflection to his commitment, actually, in organizing this webinar. I wholeheartedly thank the GIET management and Dr. Dhanraj sir, principal, Dr. Ramachandran sir, vice principal, for their emphasis and support for this connection of this webinar. I also extend my thanks to Dr. Sundar and Dr. Vijay Lakshmi for their extragenous support. I extend my special thanks and gratitude to Dr. Jan, who is a technical supportive during this entire webinar. I thank the entire crew of GIET School of Pharmacy and also Vikas Institute of Pharmaceutical Sciences for their students and persistence support towards this webinar. It could not be possible without thanking all the registered delegates who have believed us and they have participated in this productive webinar. Thank you all for your critical preparedness and readiness response actions in the spirit of solidarity. Thank you, everyone. And all special thanks to Dr. Praveen. Really, thank you. You have done a great job.
Sure. Thanks again.
Thank you, madam. I thank once again Dr. Praveen and Dr. Sumalata, all the panel members, and all the participants for participating in the international webinar in cancer immunotherapy. Once again, I thank everybody. Thank you very much.
Thank you,
Thank
sir.
you.
Thanks everyone.
Shall
Good
we-
luck. Bye.
Thank you. Bye.
With this,
Shall
we
we sign
are
off, sir?
ending the session. Thank you very much.
Thank you, sir.
Yes, ma'am. And the feedback form will be posted, is already posted in the chat box. All the participants, you can send your feedback form, and the link is posted in the chat box. Thank you very much.
Thank you, sir.